Hi, everybody. Good to see you. It's my pleasure today to introduce Dr. Cody Garrison. He recently joined my group this past September. Prior to arriving here, he just finished his PhD last year at East Carolina University. And you'll hear about some of his dissertation work today. Before that, he got his master's at Old Dominion University, where he also got his BS a while ago. Cody has come here to look at microbial communities that colonize microplastics out there in the ocean. And so he's got a study underway to do metagenomics of communities of, we hope, plastic degraded microorganisms, and we're looking for genetic signatures of that in the coming months. He has some previous experience looking at biofilms and the built environment, looking at microbial communities that colonize stainless steel. He has worked in the coastal environment as well as in Antarctica. So he has a wealth of experience that he brings here to SoMAS. I think I've said enough. Cody, the floor is yours. Handle the lights.
Sure. Want back on or back off?
Back on, that's good. Thanks, Gordon, for that introduction, and thanks all of you for joining us here today. I'm really excited to be part of the team here at SoMAS, and I look forward to getting to know all of you a little bit better, and at some point sharing my research, my current research here at SoMAS. But today I thought it might be a good opportunity to share with you a little bit about my research background and where I come from. And so this project was part of my PhD. This project in particular, what ended up being the last chapter of my PhD. It was an NSF rapid funded project in collaboration with biology, geology, and coastal studies departments at East Carolina University. And so we had the opportunity to study Hurricane Florence in 2018 and its effects on the coastal ecosystem. And so this is one of my favorite pictures from the project. Really, it's a striking image that captures the aftermath of Florence. And this is an actual quote from North Carolina Department of Transportation after the storm, several days after the storm. So we had a significant amount of rainfall throughout the state of North Carolina, and some areas reaching up to 36 inches of rain. And so as you can imagine, this had a pretty significant impact on the community. And so looking at this image of the East Coast and of North Carolina, you can see, just direct your attention for starters at the red star here. This indicates the location that the picture from the first slide was taken. So this is just to show you that it's not constrained to just the coastline. Further places inland were severely affected as well. This is about an hour drive from the coast at that location. So we know that extreme storms are likely to increase in frequency and intensity. This has been covered extensively by the Intergovernmental Panel on Climate Change as recently as last year and their sixth assessment report. And so North Carolina is a very unique area because it has this barrier island system known as Outer Banks. And within that barrier island system is contained several estuaries and a large network of inland rivers that feed into that estuary. It's also unique because, in my opinion, because the furthest east point on the Outer Banks, which is Cape Hatteras, is the closest point of anywhere else on the East Coast to the edge of the continental shelf there and the Gulf Stream. So as you all know, the Gulf Stream is an important region for primary production and balancing those carbon budgets. So any kind of events that disrupt the coastal ecosystem here in Eastern North Carolina presumably might have a higher probability of affecting those Gulf Stream environments as well. So along Outer Banks, there's a series of inlets And those inlets can essentially be viewed as conduits of both freshwater and terrestrial material being transported out into the ocean. And so what happens when you have these extreme storm events, you get a lot of inland river flooding, and those floodwaters from those rivers actually flush a lot of terrestrial organic material and microbes from the riverbanks, and all of that material goes seaward into the estuaries and into the ocean. So it's important to understand how these events might affect the coastal ecosystem and the microbial community, with the potential to impact your nutrient cycling and your regional carbon budgets. So a couple more images here, satellite images of the Outer Banks area. This is the southern portion, and the image in the top left is the southern portion of the Outer Banks and its estuaries, taken on September 3rd of 2018. And so this is 2 weeks prior to Hurricane Florence. You can see what it normally looks like. And then in the bottom left, you see the same image taken September 19th of 2018, so 5 days after Florence hit. And so right away you see the disruption of sediment within the estuary and also within the ocean there. But this doesn't tell the whole story. And so some of those inland rivers, even towards the central part of North Carolina, actually took up to 3 weeks to start subsiding their floodwater levels. And so this area is characterized as having very long residence times in these estuaries and rivers. And these all play a part in kind of a delayed or potential long-term response and affect this ecosystem. So looking back at the image on the bottom left, you can also see from some of these southern rivers, the freshwater output here that's coming after the storm. And then this satellite image from NASA showing with that freshwater, you're getting a lot of this color dissolved organic material within that. Directing your attention to the top right image, this is a photo of the Tar River in Greenville, North Carolina. And this is right next to East Carolina University. And you can see just the magnitude of the turbidity and terrestrial material that's being flushed seaward in these rivers. So of course, you're going to have downstream consequences. You're going to have eutrophication, potential hypoxia, and potential transport of pathogenic microbes even seaward into these marine systems. But these ecosystem impacts have a likelihood to be geographically context dependent. And so what I mean by that is that it depends on the proximity to urban environments or agricultural environments, whatever system you're looking at. The topography, hydrology of the region are all going to play a part in the magnitude of that impact of these storms. And so this Eastern North Carolina region is going to be impacted differently compared to Hurricane Katrina in New Orleans or Hurricane Harvey down in Texas. And so just to expand on these context dependencies just a bit further, obviously it also depends on the storm itself. So it depends on the path that it takes, the intensity, and the duration. You can see just a quick snapshot of North Carolina's most costly hurricanes and how Florence falls into that. So you can see Florence and its path that it took in orange. And so Florence was kind of unique in that once it hit Wilmington, it actually went due west across almost the entire length of North and South Carolina before it finally started to go north. And so this path is another reason why there was so much rainfall that occurred in both the central and even western parts of the state, contributing to those inland river flooding. So just to be clear here, our research questions for this project are, do North Carolina hurricanes have consequences for microbial and biogeochemical processes? And so we kind of hypothesized that we should see shifts in microbial community composition, that is the phylogeny of the community, the putative activity of that community, and also the functional potential that that community has within the ecosystem. So when we're thinking about these processes, we can start to think about them a little bit schematically to try to understand them better. And so this image tries to explain better everything that's going on when these storms hit. And we can kind of think of this as storm-enhanced microbial production. So I talked about these floodwaters and the terrestrial material, which is depicted here in this cluster on the far left. So we see this, you get your leafy materials and your more bulky recalcitrant terrestrial carbons. So these recalcitrant carbons are signified by the green carbon circles. So these are your lignins, your more difficult to decompose carbon sources. And then you also have your terrestrial microbes. Don't forget about those. These terrestrial microbes are represented by the different morphologies you see on the far left, and then also some potential nitrogen-based runoff within this floodwater as well, because we have some agricultural areas and wetlands surrounding the area. So what happens? All this material gets transported into the marine system, which you can see in the middle of the graph or figure. And these marine systems are generally characterized as being more labile, more with carbon sources that are more easily decomposed. And you also have your resident marine microbes as well, represented by the different morphologies there. So during this introduction, the terrestrial microbes, which are used to breaking down that recalcitrant carbon, are introduced to this labile carbon, and they immediately increase their biomass. They enjoy feeding on those labile carbons, in addition to that nitrogen-based runoff. That nitrogen is also labile. So they increase their biomass, and in doing so, they also create or produce these extracellular enzymes that actually allow for the breakdown of the recalcitrant carbon more efficiently. And so we can refer to this specific process as the XE2 microbial effect. Different variations of this has been given different names in the previous literature. It's sometimes been referred to as carbon priming, if you've heard of that. So if you look at the far right portion of the figure, you can see kind of attempting to quantify what's going on with bacterial production and respiration on the y-axis, and each separate individual microbial production from your terrestrial and marine sources, represented by the dark gray and the green bars, respectively, and then that additive combined XE2 effect shown in red. So you get more bacterial production and respiration, and potentially affecting that carbon budget even more without the mixture of these two communities. Okay, so Hurricane Florence made landfall on September 14th, 2018. We also had an unexpected second hurricane that hit on October 11th of 2018 that put a little bit of a kink into our sampling plan. And as I mentioned, this was a rapid funded project. So by definition, it's supposed to operate faster than traditional proposals and funding, which it did. But as you all know, it takes time for approval, accessing the funds, and general feasibility of research vessels and sampling. So we didn't get out there as fast as we would have liked to, but we did see, remember the amount of time that it took those floodwaters to decrease up to 3 weeks. Because of that, we were confident that we would still be able to see a signature from these hurricanes out in the estuaries and the ocean. And then, of course, ideally, we would have loved to sample multiple hurricane seasons across multiple years, but we were just limited to one hurricane season here and one non-hurricane season. So we were able to sample on October 26th, 27th, and November 30th of 2018, which represents our hurricane season samples. And these are represented in red. We also sampled the following year in the summer, far removed from any hurricanes, and these we considered our non-hurricane season samples. This was in July of 2019, and represented in yellow. We also had a little bit of a bonus. We had a set of samples from an unrelated research cruise that was taken on March of 2016 that took similar samples and from similar sites and were well preserved, that we were able to use just for comparison to our data to see to see what might have been going on in 2016 as well. So we chose our sites in and around Oregon Inlet. The central area here of our sampling, which is just north of Cape Hatteras, and then we tried to get samples from the north and southern portion of the estuary to get that signature from both river systems, and then along an east-west transect to the edge of the shelf, and potentially getting that Gulf Stream signature as well. So we took 63 total samples. These included surface water and sediment samples, and we extracted the RNA and DNA from these samples in order to look at the microbial community composition, phylogenetic information, and this was done using amplicon sequencing. So the RNA portion of this data actually is what allowed us to look at the putative activity of those community members. And then we also did a smaller subset of metagenomic sequencing, which allows you to look at functional genes present in your sample rather than just your phylogenetic data. And so the metagenomics would hopefully help us interpret the functional potential of that nutrient cycling that's going on a little bit better. So let's take a look at the data. This is a canonical correspondence analysis plot, abbreviated CCA plot, similar to an NMDS plot, PCOA plot, or really any ordination plot. And so the axes are arbitrary. They're just a way for us to depict the similarities and differences between our samples. So each data point represents one sample, and the physical distance between each data point is how similar or different the community composition was between those two samples. Closer together, more similar, and vice versa. We also have the unique aspect of a CCA plot is the ability to test the influence of environmental variables. And so that's signified with the arrows that are overlaid in the middle. And so the degree of correlation of these variables is represented by the length of the arrow. A longer arrow signifies a stronger correlation and vice versa. So we see that salinity, for example, the dark gray arrow, as you move to the left, salinity is increasing and your community is changing. Well, this salinity is the most influential variable in our data set. That's good. That makes sense. We know that freshwater communities are different than marine communities. But we also see a significant correlation between from the hurricane effects. So all of these samples taken in 2018, the red and the green, are significantly separated from the blue and the purple non-hurricane season samples. So this is good. This is what we were hoping to find. We want to see the difference from these two sampling periods from that hurricane. And so I'll add, we also have an overlaid aspect of the green and the red data points on top of each other, which tells us that both your active and your total portions of the community are doing the same thing. There's no difference there. So that's reinforcing. Okay, so this plot in particular is showing bacterial communities in the surface waters. But we also took sediment samples, and we want to look at the whole microbial community, which includes organisms beyond bacteria, so your archaea, for example. We're also interested in seeing what they're doing. So we can look at this 4-panel subplot of these ordination plots. And so the previous slide is shown on the top left graph, your bacteria in the surface water. And then the right column plots are your sediment communities, and the bottom row are your archaeal communities. So let's first take a look at the top right plot, bacteria in the sediment. Well, we see that the hurricane did not have an effect in the sediment community here. There's no significant correlation with that blue arrow, and no separation between those hurricane and non-hurricane data points. What we do see is a separation between DNA and RNA. So we have our blue data points and our red points, which are closely overlaid, and these stand for both hurricane and non-hurricane DNA. So taken from both years and both represent DNA. These are significantly separated and different from all the RNA samples. We think about this a little bit. This actually makes sense. Our sediment communities, we know, have a higher proportion of dead or inactive cells within these environments. We have a higher rate of cell dormancy that's occurring, higher rate of sporulation that's happening, and so just general less active cells in the sediment for various reasons, metabolic strategies, or they're just dead. So this gives us confidence that our methods make sense again, and the methods are working. So next, we want to take a look at the archaeal communities, that whole bottom row. We were very surprised to see no correlation in the surface water, similar to the bacterial communities. So why there's no difference in any of these data points in the bottom left, no separation whatsoever. The only thing that's significant is the salinity. So what's going on here? Why is there this disconnect between the bacteria and the archaea? So you get to thinking, we know that archaeal diversity in general is lower in terrestrial soils than in marine systems. Okay, so maybe there's just, in general, fewer cells getting flushed seaward in these floodwaters. That makes sense. A couple other theories could be that bacteria may be just more adapted or well suited for handling these potentially eutrophic hurricane conditions. Alternatively, archaea may be characterized by lifestyles that are unaffected by hurricane conditions, such as chemoautotrophy, not interested in the organics. So we're excited. We're getting more evidence about what's really going on during our hurricane season in 2018. We also have another theory. Maybe these sediment communities and archaeal communities may be more resistant or resilient in general to hurricane conditions, which could be an exciting area of research for some future grad students. Okay, so whenever you're analyzing a microbial community, it's always important to look at your diversity indices. And so diversity indices include species richness, species evenness, and some measure of diversity. So in this case, we use Shannon diversity. And so just to clarify here, species richness is the sheer number of species within your community. Evenness is how evenly their proportion or abundance is distributed. And then diversity is actually a combined measure of both richness and evenness. So looking here, we see two plots, evenness and diversity. And these are all of our samples, box of whisker plots, where our red boxes show hurricane condition, and our sites are broken down by shape. So our circles are estuarine sites, and triangles are marine sites. So marine sites, in this case, are anything greater than 25 parts per thousand. So we see on the x-axis our samples separated by sediment and water. We see no significant difference in the sediment communities as far as evenness goes. And then looking at in the surface water, though, we see start to see this difference in evenness. We see this lower community evenness significantly occurring in the hurricane condition samples, but only at the marine sites and not in the estuary. And we see basically the same thing happening in the Shannon diversity. We have a little more variability, but the same general trend is happening in the surface water for the Shannon diversity figure on the right. So we have this disconnect now between our estuarine sites and our marine sites during hurricane season. Potentially, this could be due to slower advection of nutrients offshore or a faster rebound of the estuary to pre-storm conditions. All right, so we have this unevenness at these marine sites. So what could that mean? Well, a higher evenness means a more even community, more equally distributed as far as abundance and proportion go. Lower evenness, maybe you have some dominant taxa that are just outcompeting the other taxa and making their competitors more rare in abundance. So we want to investigate that further. What could be causing that lower evenness in the community? And so before we do that, though, I just want to show the data for species richness, which is shown in the far left column, and then RNA, all the RNA data is on the bottom row. So the previous slide just showed DNA, evenness, and diversity. We see that there's no difference in species richness in sediment or the surface water. And then your RNA samples, again, agree with the DNA. So your active members are having the same trends and supporting that DNA data. All right, so what's driving this unevenness in the community? Well, one really powerful tool to look at that is to look at your copy number of the ribosomal RNA. So this is all of this data so far that I've shown is amplicon sequencing data, which is processed by sequencing one gene, the 16S ribosomal RNA gene. And so you can look at the copy number of that specific gene, that specific ribosomal RNA, as a proxy for metabolic lifestyle in your sample or your community. So in general, communities that have an average higher copy number of this gene suggest a more copiotrophic lifestyle. So they prefer higher nutrient conditions as opposed to a lower copy number, as more often found in oligotrophic organisms, preferring those lower nutrient conditions. So we were excited to see this really big significant difference, again supporting our previous results of those hurricane condition sites, not only the marine sites, not in the estuary. So this suggests a higher abundance of copiotrophs that may be driving that unevenness in the community. And then, not to be understated, this also just reinforces the fact that these floodwater nutrients have the capability to reach distances offshore, those further marine sites. So you always have to look at taxa plots. You have to look at taxa plots because you want to know exactly what taxonomic members are driving these changes. We're looking for copiotrophs. We're looking for that unevenness in the community. So this figure shows marine sites specifically, and the average percent relative abundance of these microbes across different taxonomic classes. So classified to the class level here. And so just looking at the most abundant organisms with the biggest significant differences, we see flavobacteria are higher in abundance during hurricane season compared to non-hurricane season. These are common heterotrophs that are capable of degrading both simple and complex carbon sources, so your labile and your recalcitrant carbons. Okay, that's good to see. That supports what we found. We have a higher abundance of gamma proteobacteria. These have very diverse roles, which makes it challenging to interpret. But there are a lot of members within the gamma proteobacteria that are copiotrophs. And even some of them have been shown to be resilient to disturbance as well in previous literature. So that's interesting to see. And then finally, we found lower abundance of alpha proteobacteria. So its most abundant member, can anyone tell me what the most abundant member of alpha proteobacteria? It's your oligotrophs, your SAR11, the most abundant bacteria in the ocean. So these are, as I said, oligotrophic organisms that are decreasing in abundance during hurricane conditions in these marine sites. So this corresponds to that increase in copiotrophs, decrease in oligotrophs, and that disruption to the community. So we have our estuarine sites that we have to look at. We see a higher abundance of planktomycetia, which are also copiotrophs. We see a higher abundance of stringobacteria, which degrade specifically complex carbon sources, your recalcitrant, for example, cellulose, chitin, they've been known to degrade. And then finally, a higher proportion of beta proteobacteria as well. And these are known freshwater inhabitants, which support not only the nutrients being there in the floodwater, but just the salinity being much lower as well, and lingering after these storms. We also found an increased abundance of pathogenic indicator taxa in all estuarine and marine sites during hurricane season compared to non-hurricane season. And so these, we found Legionella and Prevotella. These are classified or represented based on genus level classified organisms. And so you can see pretty striking significant difference, at least to me, the difference in relative abundance during hurricane season, all hurricane season sites. And so this needs to be made aware of. Obviously, can have potential human health risks, recreational aspects for the whole Outer Banks area during that hurricane season. Okay, so as I said, this was a collaborative project, and our collaborators in the geology department focused on the geochemical variables. They measured, for example, total suspended solid, dissolved organic carbon, and total dissolved nitrogen. And you can see those represented in these figures here. We see our white symbols representing hurricane season 2018, and black symbols representing the following year, non-hurricane season. Circles representing dissolved organic carbon, triangles are your nitrogen and on the right figure. And we were really disappointed and frankly surprised to see that we didn't see any correlation or trend in your geochemical variables measured here with the hurricane season. So we're still trying to figure out even now why this is, but the most likely scenario is that we missed the nutrients. We sampled too late. The microbes may have a longer residence time in these estuarine marine systems in eastern North Carolina than the actual nutrients that they consume. So that's one line of theory. Another option is that the different size filtration that was used, the geology department used 0.7 micron filters for their dissolved nutrient work. We used 0.2 micron filters for our microbial work. So anything that might have passed through that 0.7 filter could potentially influence this microbial signature that we found so far. And then finally, water quality variables that were not quantified could have also influenced our data set. In my opinion, the most likely scenario is number one, we just missed the nutrients. You can see a couple of really low salinity sites that did have high nitrogen during hurricane season here. But other than that, you can stare at these figures for a while and you just can't see much of a trend of anything. No difference. All right, so I had to mention these geochemical variables, because I know everyone is probably interested to see how they compare to the microbial data, but I need to get back to the microbial data. So that's the important, what I'm interested in. So to this point, I've focused just on amplicon sequencing results, which is just your phylogenetic data. But I also did, we did metagenomic sequencing to look at all of the functional genes present, total genes present in your sample. And so whenever you do metagenomic sequencing, you get an absorbent amount of data, very, very large files of data. And so you basically have two strategies to analyze your data. You can either know exactly what gene you're looking for, or you can kind of just throw everything at the wall and see what sticks. This first graph is the former. We're interested right now to see the presence of lignin degradation genes specifically. So these can be kind of a terrestrial marker representing the presence of these terrestrial microbes that are capable of breaking down those recalcitrant carbons. So more specifically, we were looking for your lactase, your peroxidase genes that are able to genes that allow for lignin degradation. Another thing that I have to say is that metagenomic sequencing is very expensive, so you're limited to a smaller subset of samples here. So we could only sample or sequence 10 samples out of the total 63 for metagenomics. So you lack that statistical significance, unfortunately, but you can gain some really strong insights from your data set. So we did see a higher abundance on average of these lignin degradation genes at our lower salinity sites in both the sediment and the surface water. And so this supports just the general persistence of these bulky terrestrial carbon sources lingering around out in the storm. So this slide is kind of showing more of the throw everything at the wall variety. And so you're able to look at all the potential metabolic pathways, or all the more common ones at least, and whether or not they might be present or absent in your sample. And so we did this using a program called DRAN, which stands for Distilled and Refined Annotations of Metabolism. And so this is a lot to look at. I'm going to focus on the most important stuff in a second, but just to show you, your columns here represent samples, and the color gradient represents percent completeness of these different metabolic pathways. So if all the genes are present for that particular pathway, you're going to have a dark blue symbol, and if it's 0% completeness, it's going to be your pale yellow. And so the most interesting results that jumped out at us is this subset here. So again, your samples are on your columns here. The far right sample is sediment. So that's always going to be different than your surface water. So it's kind of a reference, but not really comparative. The far left column here is our hurricane season marine sites. And that's where we saw the biggest difference between those, again, similar to our previous results that I've shown, difference between those marine sites and the rest of the data set. So we found less genes related to glycolysis and citrate cycle pathways. And these are pathways for the breakdown of your labile biopolymers. Presumably this decrease could be corresponding to a potential increase of the breakdown of more recalcitrant or less labile polymers. We also found less genes related to the glycoxylate pathway. And so this pathway is often used as a proxy for SAR11 abundance. It's a metabolic strategy that they take in oligotrophic conditions. They use acetate as an intermediate carbon source to give them an edge over their competitors during oligotrophic conditions. So we found less genes for that. We also found less abundance of alpha proteobacteria. Results are complementing each other, supporting each other. That makes us feel good. So we have decrease in oligotrophs, increase in copiotrophs. We also have less genes for reductive pentose phosphate cycles, which is important for photosynthesis and primary production. Okay. Remember, this is a marine site close to the edge of the continental shelf. Very important region for primary production. We're seeing reduced primary production, increase in heterotrophic metabolism, those copiotrophs, and that potential to disrupt the carbon budget there, potentially switching from net autotrophic to net heterotrophic. Maybe a stretch there, but it's possible. We also found less genes for 3-hydroxypropionate, which are genes for photosynthesis pathways as well, specifically for carbon fixation. There's plenty of other pathways that you can search for using this program, and this just shows whether it's present or absent. So green being present and light blue being absent completely. Again, focusing on just the most important pathways that stood out to us were the nitrogen metabolism pathways. And so you see just kind of a general increased diversification of these nitrogen pathways in both marine and estuarine sites during hurricane season. So you have specifically increased abundance of ammonia oxidation genes, nitrate reductase genes, and nitrogen fixation genes. And so this kind of supports all that nitrogen-based runoff being an important factor in those floodwaters and input into the system. So thanks for bearing with me here. I just want to summarize. that these, in my opinion, these ecosystem impacts are geographically context dependent. And this is important because every storm and every ecosystem is going to respond differently. And it's important that we know how each system behaves. So your proximity to your urban or agricultural environments, topography, hydrology of the region, all going to be important in addition to the storm characteristics itself. We found contrasting correlations across habitats and domains. Probably the most exciting result to me was that difference in your bacterial and archaeal communities and in the surface water and the sediment. So maybe a potential increased resistance or resilience of those sediment and archaeal communities during hurricane season. We found a decrease in community diversity and evenness, increase in copiotrophs and pathogenic indicator counts. And then it's really important for us to understand how these systems are impacting the area so that we can start to predict and plan accordingly for the future if they are more frequent and more intense, and how they might affect our nutrient and carbon budget. And with that, I'd just like to acknowledge my wife and daughter, who are always my #1 supporters of my research, and all the folks at East Carolina University and our funding agencies. And thanks to all of you for being here today. And with that, I'll take questions. Yes.
Hey, of course, my question is about fungi. Or whatever, I can spell it, it's only 5 letters. But what does it say? 5. But certainly terrestrial detritus is likely more often than not dominated by fungal degradation, as well as surface soils are either half or sometimes more fungal.
Okay.
And the only other thing I know is that 16S RNA doesn't give you any information about eukaryotes and fungi.
It does not. No.
Did you end up with any information from your other analyses about changes in fungal?
So the metagenomics would sequence all the genes, including eukaryotes. So your lactase and your peroxidase could have potentially come from fungi. Yeah, we did not do amplicon sequencing on fungi or the ITS region, which would be for fungi. But the metagenomic data could apply, yeah.
Oh, interesting. So you can't, so there's not a distinguishing type of lactase genes that are some for fungi,
Specific for
some for
certain
bacteria.
domains or taxa. Yeah, it's a good question. I'm not aware of what that is. Yeah.
What a question from the students. From each student. Let's start with one.
Yes.
Do you think if you lagged your sediment sampling behind your surface water sediment or surface water sampling that you would have seen a hurricane effect a little bit more?
That's a good question. Potentially the settling of these organisms over time. It's possible. I guess I hadn't really thought about it. I was always a little bit. I always wanted to go sample faster than we did, so I wasn't thinking about what could happen even longer. But yeah, it's definitely possible. Good question.
Yeah, I was wondering if you might be able to see that, like since you had so many sites that you sampled, like there were probably days in between when you did your sampling, right?
Yeah.
Like I wonder if you could compare like the sites that you sampled first and then see that lag a little bit more like with these other sites. But I don't know if it's like, if you could say it was like dependent or.
Yeah, that's a good point. It's something worth looking at. Thanks.
Yeah.
Yes.
I remember there was like a second hurricane at Florence. Would that have affected your data at all, or was it just like hurricane season?
Yeah. So initially we were hoping to just focus on the effects of Hurricane Florence on this community, and then there's a second one that comes, and we actually didn't sample until after the second one came. So we're looking at a sequential storm event, right? So the effects from 2 sequential storms, which maybe that's going to happen more frequently in the future. But yeah, not sure if it's possible to tease apart, what came from what. It's kind of just, we're trying to sell it as just during hurricane season, this is what's going on. Yes.
I'm not sure if I heard correctly. How can after hurricane?
So the Florence hit on the September 14th. Michael was on the 11th, and we sampled October 26th, 27th, and November 30th. Yes.
Do you know much about the potential for a storm surge to actually penetrate into the estuary region, or are the barrier islands too high, too solid for that to happen?
There's definitely constantly new inlets being formed and being repaired constantly. So I think some of these down here were definitely new, or at least got larger. I'm not sure exactly, how much impact from the storm surge from the ocean side. I was kind of focused on that delayed river surge of fresh water after the fact. But there's all sorts of different complex components to these events. That's a good question. Did I answer the question, or was there?
Yeah, I mean, I think it could be lots of other questions to follow, but I think that's it.
Yes.
So how much do you think overall this land to coastal ocean interaction, even if periodic, contributes to the overall productivity offshore?
It's also a good question. It's hard to say. I mean, technically, with every tidal cycle, you're getting freshwater and terrestrial material going out, but it depends on the magnitude, right? So you just have so much more material during these disruption events that have a higher potential to impact the community than what they're normally used to.
So I don't remember seeing that comparison, or do you have, this is what you normally
What
see,
normally see?
this is what you see.
Yeah, we try to avoid. Well, we've gotten a little bit of pushback from trying to say it's like a time series, situation. We're trying to say this is what happens during hurricane season over before and after the rebound. We're trying to focus on just the impacts of those events rather than just the normal. But presumably the 2019 samples should be a baseline. That's the goal. Yeah, I think, hold on one second. We have another hand to that.
I just missed that part, but you might just clarify why that DNA and RNA is more correlated in the more positive.
Why the hurricane effects were seen more in the water column than the
No, why
sediments?
the RNA and DNA?
Why the RNA and DNA were different in the sediment? Or okay. Yeah, so this figure in the top right, you can see all of the DNA are overlapping here, the red and the blue data points, and separated from that RNA. And so our reasoning is that there's a higher proportion of dead or inactive cells that subside or reside in the sediment compared to the water column. Water column has a lot more active bacteria and microbes in general, where you have different life strategies such as dormancy and sporulation to kind of resist those harsh conditions of anoxia, low nutrients, just unfavorable conditions in general. Cells might go into dormancy or create spores within their cell membranes to kind of as a nutrient source or to resist desiccation. Yeah.
How do you think your data would reflect a biological accumulator like a bivalve? Do you think it would show similar results to the water column, the sediment, or would it be faster, slower moving? Does that have strong implications for food safety standards?
Yeah. That's an interesting question. Definitely haven't thought about it before. I mean, Yeah, so it's kind of a contained microbiome within that organism. But I mean, it filters water pretty rapidly, right? So it should have a high turnover rate. So I don't see why it wouldn't be any different than the surrounding seawater, in my opinion.
They're great integrators of toxicants over time. Process.
Yes.
So I'm curious about like how long term these effects are. So you looked at like within a month or two, right? With this data?
Yeah. So our last sampling date was November 30th. I mean, if you want to talk in terms of the first storm, Florence, that's almost 2.5 months that we still saw some effects.
What do you project, or would you think that you would see stuff even further out than that, like a year, 2 years? Like how impactful are hurricanes in relation to like long timescales?
Well, also a good question. We know that they're different come July of 2019. We know that much. So, I mean, we don't really have like a set rainy season, I guess, other than just hurricane season. So it's going to vary over the years, depending on how much precipitation you have and how much nutrients are going into the water over the winter time that are affecting that that signature over time. Or if it's a dry winter, then that could bounce back faster, for example. So I think it's going to vary every year depending on what's going on. Yes, Gordon.
Cody, I think I missed something in the CCA plots and the box and whisker plots, all of those, where you compare DNA and RNA. What is it you're actually comparing? You've got the ribosomal RNA gene for your DNA. What's the RNA?
So we actually, I didn't touch on this, but we used a technique that's really cool. You extract RNA and you reverse transcribe that RNA to complementary DNA, cDNA, and then you just do the amplicon sequencing of that cDNA. And that gives you your phylogeny data for which community members are active, because it came originally from your RNA, and you can compare that to.
But it's, and again, it's 16S gene.
So again, 16S, correct.
Maybe for the benefit of the students, you talk about putative activity.
Yeah.
Using RNA, you want to explain what you mean by putative activity?
So putative being just kind of a safeguard word. We're not proving the function is happening. So to do that, we would need to do metatranscriptomics. So similar to metagenomics, which is sequencing all the functional genes and the DNA based on DNA. Metatranscriptomics would be sequencing all the genes based on RNA to see those active ligand degradation genes are actually active, and that function is actually occurring. Whereas here we have just the activity of that taxonomic group. We know that this organism is being active. We can't say for certain that it's actually degrading the ligand. So that's why we say putative activity. Yes.
I'm curious if the magnitude of like the delivery of the guys that you looked at specifically, and I guess probably sediment and nutrients as well, if that magnitude is proportional to like the quantity of rainfall. So say you have a hurricane that delivers 12 inches of rain, and X amount of delivery. You have a hurricane that delivers 12. For 24, you expect 2 times the amount. I guess that also begs the question. Is there like a recovery period where if you have a hurricane that immediately follows another quick hurricane, is the delivery the same? Is it less, or is it I guess the terrestrial system is able to like recover more biomass that that can then get dumped back into the system or more? If that makes sense.
Yeah. So there's a lot of different ideas there.
Yes.
So it depends. All the characteristics of the storm is definitely very context dependent. And Florence was unique in that it started out as a category 4 or 5 out in the ocean. And once it made landfall, was actually down to a category 1, and it decreased its speed tremendously. So it moved extremely slow over the state, and that's why the biggest impact was just pure rainfall. So if you had a stronger, faster moving storm, then you can actually start to look at maybe the winds and the wave action and actual physical disruption more so than just rainfall, which would be a different aspect to look at. And then Hurricane Michael, which occurred a little less than a month after. I would think that if the system is not fully recovered before Michael hit, based on how slowly the rivers subsided, in my opinion, that kind of delays the process. And we thought it was a kink in our sampling plan, but it may have actually helped delay seeing these microbes in this hurricane signature too for us. Did I leave anything out?
Well, in that case, I think it's time for lunch. Let's thank our speaker one more time.